Welcome to the second part of the introductory lecture to medical research. This is a diagram that summarizes the types of studies we saw in the first lecture. The first type of study we see here is a cross-sectional study which shows individuals who were examined at a single time point. The second type of study we have on this diagram is a case control study wherein you have two groups, the case group and the control group. And these are individuals with a particular outcome in case of the case group, and then individuals without a particular outcome in the control group. And they were looked back in their history to see whether they had any factors present or absent. So that's in terms of their exposure. In terms of the cohort study, we saw that you had individuals who were exposed to a particular factor and individuals who were not exposed. So the exposure was the starting point, and then eventually they were followed through to see whether they were able to come up with some certain outcomes or not. So basically, this is a summary of the different types of studies we saw in the first lecture. The case in most researches, we cannot carry out a study on a whole population. So the target population is identified and a sampling is done in order to determine eligible representative candidates for that research. So sampling methods are ways to select a sample for the data of a given population. It is unrealistic to want to collect, as mentioned, data from the entire population for various reasons. Firstly, the population would be too big, it may take too much time, it would cost too much money, and therefore it would not be something that would be practicable. So as a result, there are different sampling types or methods that are used when we're doing research studies. One of the most common one is the simple random sampling, wherein we gather representative samples from a population where each member of the population has an equal chance of being selected. The second type would be a stratified sampling, where a smaller group or strata within the samples are represented proportional to the population. An example of this would be sort of having in a classroom maybe of about 50 students, you would maybe have 10 females and 40 males. So you can use a stratified sampling size method rather in order for you to be able to make sure that you have a proportional representation of that class population. A systematic sampling would involve every member of the population being given a particular number, for example, and after the first member is chosen at random, then the remaining members are chosen at that given interval. So for instance, you can say that you're going to choose a particular subject or candidate, and then eventually every other third candidate after that. So in a systematic manner, you would be sampling the population. The other type would be a cluster sampling, where clusters are used. And also you can have a non-random sampling, which would be, for example, a convenient sampling, which is used for the ease of data collection. So for instance, in a classroom again of 50, instead of going randomly and selecting candidates, perhaps for ease, you can choose the first 5, which is not random, and it may not necessarily represent the whole class population. So there are different types of data. Primarily, data can be subdivided into different categories, but you can have what's called primary data. So that's data that's collected from the original source. And then you can have secondary data. So that's data that's collected from a secondary source, meaning not directly by the researcher. You can also classify data into qualitative, so that's non-numerical data, or it can be qualitative data, so that's numerical data. And qualitative data can further be subdivided into discrete data, so that's exact values or whole numbers that are not rounded, or quantitative data can be continuous data, that is measurements that can be rounded. So this is a diagram, again, showing you the different types of data. Primary data that's collected and handled and analyzed by the individual or the researcher collecting. Secondary data is often data that somebody else has collected and it's being analyzed now. We did mention qualitative data, which is non-numerical. So examples would be things like color, days of the week, brands, or animals. And then numerical data would be quantitative. In other words, it can be either continuous, so that's quantities that can be rounded. So things like time, length, mass, capacity, and discrete data would be exact values. So these are numbers of maybe students in a class, the shoe size, score rolled on a dice, and the frequency of events that occur. Here would be this would be dealt more in detail once we come to the module on biostatistics or statistics in research. So basically, just to talk briefly about what sensitivity, specificity, and positive predictive value and negative predictive values are. So basically, these are diagnostic tests that are used quite frequently in statistics. And basically, on the columns, you have the presence or absence of disease, and on the side of the rows, you have a positive test or a negative test. And eventually, this would be used to calculate your sensitivity, specificity, positive and negative predictive values. Research ethics, why does it matter? For a host of reasons. But the most important ones are firstly to protect the participants and ensure their safety during research. Secondly, would be to maintain the scientific integrity of the research and the research process itself. Also to uphold human right and dignity, as this is quite important. The whole essence of research is in order to contribute to science and make sure that we advance healthcare and scientific knowledge as a whole. Another reason for following research ethics is to ensure social responsibility, to help build trust in research and institutions, and obviously to be in compliance with legal and regulatory bodies. So these are just a few reasons why research ethics is quite important. It's often a neglected portion of research, but this is actually what guides research and determines the rules when it comes to research in human subjects and in other fields as well. So you've got your research findings. How do you communicate your research findings? And this is quite important. There are various ways of communicating your research findings, but we would focus on the most common ones. There are various ways of presenting your research findings and writing a scientific manuscript. They all have their nuances when it comes to the manuscript writing, but we'll just look at most of the commonalities within these scientific writing. So basically, these are some of the parts of your research paper. You obviously would have to have a title, an abstract, an introduction, literature review, that may or may not be an entire section on its own based on the type of manuscript you're writing, the methods, the results, the discussion, acknowledgement, and reference. And we'll look at each of these different sections in detail. So as mentioned, your scientific manuscript is a way of presenting your findings. So it can be an article in a peer-reviewed journal, it can be a thesis presentation for a dissertation, for example. And this is often a highly structured document, so there are rules and regulations guiding on how you write a manuscript. And there are slight variations depending on the type of manuscript you're writing. What's common to most of them, especially when it comes to peer review articles, there's an acronym called IMRAD. So basically, IMRAD is you have an introduction or background part of your article as the first, and then you have your material and method section, you have your results and discussion, and eventually your conclusion at the end. And it's important to note that part of your manuscript you would have an abstract. So basically, an abstract is a summary of your manuscript, which involves each of these subsections that I just mentioned. And basically, it's important to note that your abstract would be the first port of call, or that would be one of the first things that any reader would come in contact with if they want to sort of access your or read your full article. So when you do searches online, you would often get not just the title, but most often you get the abstract to give an idea of what's exactly is in the full manuscript. So it's important to make sure that your abstracts are reflective of your actual work. So the title, that's the first part. I mentioned that it is actually a very critical and important portion of your article or your scientific writing. It is one of the things that would pop up when you do a search. So it forms the first impression of your writing. It also helps to increase discoverability. So if you have a title that actually makes it easier with certain keywords, then your article will be easier to find. It provides clarity and focus on what your article or your writing is about. It helps to attract audiences and readers, and it establishes relevance if your title is comprehensive enough. The introduction or background. So this is the portion that lets the reader know the background of your research first. So the main objectives of your study tend to come out or are actually emphasized in the background portion of your writing. Things like why was this study done? Why does this study matter? In what context was this study done? And what's the primary hypothesis? What question are we trying to answer with this research? So generally, the introductory or the background portion of your write-up would include three subsections. So the first part of your introduction would have to provide background information. So this is context for research. It discusses the existing knowledge and the literature that's related to that particular topic. The second portion would be the problem statement or the research question. So this clearly articulates the issue or the question that the research aims to address. And then the third part would be the objective or the purpose of your study. So this outlines the specific goals and aims of the research, including what the research or your study intends to achieve. So your methods and materials section would be a crucial part of your study. It should allow your readers to be able to evaluate your work. So that's to say that they must be able to describe all the aspects of the methodology that you used in your study. They must be able to replicate that study if they do desire, because you would have provided all the instructions on the methods that were used to carry out that study. So a good method section would leave no questions about how any of your research work was done. So the results portion of your write-up would be purely on data. It tells us what the measurements were, so there are no comments. There are no opinions that are given on your results. It's just your facts, your hard facts. Usually there's a table 1 that is in the results section. So the table 1 traditionally in almost all medical manuscripts describes your population that you're studying, what the characteristics of that population who were enrolled in that study are. So the next portion would be the discussion. The discussion is an important part of your write-up in that it helps to describe your research findings. It helps to make comparisons between the results you found with existing scientific knowledge. It's a subjective portion of your write-up, and it helps to discuss the outcomes that were found and how and why your study has been able to bring some new knowledge to existing scientific understandings of a particular topic. So the other two sections would be the acknowledgement and the reference section. Acknowledgement, as the name implies, basically gives an idea of the people or institutions or organizations who added their contributions to your work in a successful manner. And the reference would be a citation of the reference articles that were used in your paper in a specific format. There are various formats. One of the formats that are used in reference would be the Vancouver format. So your reference would include name of author, article title, name of the journal, the volume number, the year of publication, and the page number. As mentioned earlier, your abstract is a short version of your paper, giving just the highlights of what's in the actual paper. And it needs to be as comprehensive as possible in order to be able to attract readers to want to read your whole article. So we've seen the research process. This is just a diagram summarizing that process wherein you define your topic, you narrow it down, and then you gather background information to see what exists already in the scientific literature and what your study would be able to bring about. And then in that process, you create a research question. And once you've created that research question, you start working on your write-up so that your thesis statement is formed, and then you find and evaluate different sources where you can get information on existing scientific knowledge on that topic, and then those sources are cited, and eventually your paper is written. Once the writing process is over, it becomes now a question of trying to disseminate these research findings. So most often you would do a publication in order to share your research findings through either peer-reviewed journals, or it can be a thesis in terms of the students in medical research. And there are various ways of disseminating information in order to allow the scientific community to be able to build upon the knowledge that you have created in a similar way that your research relied on writings or scientific manuscripts that were written prior to your research. Another way of disseminating your research findings would be through a conference presentation, wherein you have the opportunity to present to other scientists or other medical personnel or other researchers and be able to receive feedback on your work, and also allows you to be able to collaborate and network with colleagues. You can also have public outreaches where you engage with the general public through the media, podcasts, or educational initiatives that helps to create awareness and promote the impact of your medical research. Clinical implications, especially for translating research findings into evidence-based clinical guidelines and practices, is essential for improving patient care and outcome. So this brings us to the end of the introductory lectures on medical research, and in subsequent lectures we'll be delving more in detail on some of the concepts that we've already seen in this introduction, but a lot of new concepts as well as medical statistics, in order to enable us to be able to not just read and understand medical literature, but also to be able to write and produce scientific material of high quality. And thank you.